Alright, we're looking at Unit 3, Lecture 3, Overview of Immunity. So in the next couple chapters to round out this semester, we're going to be taking a look at the immune system. And here we're going to kind of begin with an overview to kind of give you a broad perspective of how it works. And then in the next lecture this week, we're going to drill in on what's called innate immunity in a little more detail. So, to begin, we're looking at chapters 15 and 16. And in a broader sense, we're looking at immunity, but we can also refer to it as a defense system. I'll kind of elaborate the difference here. It's ultimately the defense system that keeps us alive. And in terms of infection and disease and even cancer, all of these things are kept in check by a properly functioning immune system. So without it, we wouldn't be here. And it's literally the difference between life and death. In chapter 15, the main focus is on innate immunity. And the idea of innate immunity is that you're essentially born with these characteristics. And so long as you get the right genes, which most of us do, everything is kind of set up from the time you're born. In chapter 16, the focus is a little more nuanced, and this is what's referred to as acquired immunity. And the idea here is that you're not necessarily born with this defense. You have to bring it in to existence through exposure and everyday interactions with microbes. So when you hear the phrase building your immune system, they're actually referring to the acquired immunity, which is also sometimes referred to as the adaptive immune system or adaptive immunity. Now, you're born with the underlying mechanisms to acquire or adapt, but you actually have to bring that out through exposure. In other words, it's not quite ready to go until you start interacting with microbes in the real world. So that's kind of the difference between the two. And because there's enough detail for each, we'll kind of take each one in a separate chapter in a separate week. So let's start with a broad overview. Here what we're looking at is what we call the defense system. So part of why it can be referred to as the defense system, and then in other contexts the immune system, is there are three levels to the defense system. And the first one is not technically part of the immune system. It's a bit of a semantic argument, but that first line is what we call barriers to infection, or at least that's one of the ways it's commonly referred. And this is really more of the anatomy and the physiology of the body. And it doesn't technically constitute the immune system, but it does make up part of the defense system. And that's kind of where that term defense versus immune differentiates. A little semantic, but that is the difference. The second line is where the immune system begins. And this can be referred to both as innate or non-specific. And we'll describe why that is as we go. The third line can either be thought of as specific or adaptive and or acquired. So barriers, first line, non-specific, second line, specific, third line. Okay, so the first and second lines technically together make up this idea of innate immunity because you're essentially born with those characteristics. And the third line by itself is what we call the acquired or the adaptive immunity. Because, as I mentioned, you have to essentially bring that online through exposure in the everyday world. Now, this figure here gives you a little more detail. So, broadly speaking, defense, innate, first, second lines, acquired, third line. And then they branch down further so you can kind of see what general characteristics make up each. So first line would be things like physical barriers, skin, mucous membranes. Our microbiome actually plays a role in this. If you remember, I talked about the concept of microbial antagonism in one of the lectures last week. That kind of acts in a certain way as a line of defense. And then chemical barriers like saliva, stomach acid. These are technically part of the first line as well. And almost all these do other things besides just defense. Your stomach acid helps you digest, for example. But in this context, they can also be thought of as a defense. Second line, a key part of the actual immune system. This is where we see cells like phagocytes, processes like inflammation, fever, and a more nuanced discussion called antimicrobial products. That one is probably one of the more difficult to really get through all of it, but we'll kind of touch on a few examples there. When you get to the acquired or third line, this is where specific cells called B cells and T cells come into the equation. And B cells produce antibodies. So if you're familiar with antibodies, those are a product of B cells. So all of these things end up playing an accumulative role in the immune system or the defense system, but each kind of have their own little place within the grand context. Now, we won't get into this here, but there is also an indication here that some aspects overlap. Gamma T cells and natural killer T cells sort of fall in between these two. Can't quite place them in one or the other. But generally speaking, we're going to focus on kind of each defense by itself there. Now, the truth is, these don't operate individually. So we can kind of characterize each one, but they all play an accumulative role together to keep you healthy and free of disease. Without these overlapping aspects, none of them really by themselves would be sufficient. So you have to kind of understand how each works, but then also understand how they play a role in the bigger picture. So, for example, one of the important aspects of a barrier to infection would be that it's made up in areas of the body of mucus. So this, for example, is found in the lungs, in the gastrointestinal tract, the stomach, even in the reproductive organs, down inside the body, on the internal areas. On the very outer layer within the body, so it would be the inner part of the body, kind of in the cavity, like the cavity of the gut, you have a little layer of mucus that lines all the organs. And it essentially acts to trap microbes to keep them from just moving throughout the body freely. So we call that a first line. But as you age and as you get exposure through everyday interaction, that mucus eventually builds up antibodies that are a third line mechanism but overlap with the first line. So the mucus first line over time develops the presence of antibodies, a third line, that work together simultaneously. So that's one example of how they overlap. Another example would be macrophages, which are considered a phagocyte, part of the innate or second line of defense. And these cells by themselves can play a defensive role in the second line independently. But at times, and actually commonly, they will also play a role in activating lymphocytes, which are B and T cells. It's a little bit of a broader term that kind of describes mostly B and T cells. So they actually play a role in activating those cells in many cases. So that's another example of where the two overlap. And another one would be complement proteins. These are an example of the antimicrobial proteins. They play a role in enhancing the effect of antibodies. So complement proteins, antimicrobial, second line, enhance the activities of antibodies, third line. So those are just a few examples of how the body ultimately has its own sort of characteristics in each line, but overlap to play a larger, broader role. And real quick, what I want to do is just kind of illustrate that to give you a quick visual with not every little detail. But another way to think of a barrier is as the skin. So if this is the outer surface layer of our skin, the outside world, inside of our body rather, the skin acts as a fairly nice barrier that all by itself is defensive. But if we get a break in the skin, a cut or something along those lines, suddenly that barrier has been bypassed. And now we have an opening in the skin that can allow microbes to come through. So we have now kind of a barrier, a breach in the barrier, so to speak. So if microbes are normally kept out, all of a sudden they're coming in through a cut, for example, and these represent microbes. So they come down inside the body. So normally the first line, skin as an example, keeps them out. now all of a sudden they get through it. What you'll find below the skin in every area of the body are cells like macrophages. And macrophages have this ability to recognize these pathogens with little receptors that are found all around the outside of their cells. And ultimately, those little receptors can recognize the microbe and grab a hold of it and bind to it. Now, what can happen there will be a couple of different things. Number one is they can send out a chemical signal that they produce in the cell and then secrete into the environment, and that can then activate more cells of various types to come in response to this signal. So the signal activates more cells, and then they migrate to the area to help that macrophage in dealing with these microbes. And if the invasion is not significant, very often that second line will clear the infection all by itself. However, if the invasion of microbes is sufficient, then what can happen is the macrophage gets a signal due to the extended period that these microbes are there, and then it literally starts to move and migrate away from the site of infection, and it will travel to a nearby lymph node, which kind of look like this. And down in the lymph node, we will have B cells and T cells sort of sitting there waiting. That's part of the third line. Very often these cells are found in lymph nodes. So the macrophage travels from the site of invasion down into the lymph node, and now it's carrying that little piece of the microbe that it picked up from the site. And it will actually go and activate these B and T cells. Now there's a more detailed way that this works. I'm oversimplifying it, but the macrophage will now activate B and T cells. These cells will now start to grow in response to that activation and increase in number, and then they will all kind of collectively move out of the lymph node and ultimately travel back to this original site. And here now we'll have a greater army of cells that are all primed to respond just to the microbe that invaded. And this is partly why we call it specific. Initially, these cells kind of respond, the macrophage here, they kind of respond to anything coming across the barrier. That's why they're called non-specific. So long as it's a foreign threat, they'll respond to it. If they can't deal with it, then they'll move to a lymph node. It could be other areas. Generally, it's a lymph node. Activate B and T cells that are specific just to the microbe that they're now being presented with. They copy themselves to make lots of duplicates, migrate back to the site, and now with the specificity, they hone in on that microbe that is invading and proceed to eliminate it in a variety of different mechanisms. So those are all kind of different ways here that these systems overlap. First line, keep them out. That usually works, but when it doesn't, second line comes in. That typically works by itself to eliminate the threat. When that doesn't work, they will then trigger a third line, and that third line then will come in when the second line fails and take care of business. And so that's kind of how these all overlap. Now the third line, one more note and then we'll move on here. The third line is usually delayed. It doesn't typically respond right away. And so depending on how many times you've been infected, if it's the first time, there's a delayed response. And that can usually be 24, 48, 72 hours, or maybe more in some cases. So there's a little bit of nuance to every step here. But that kind of gives you a quick overview of how this works. So with that in mind, then, we can kind of go into some of the details here. Ultimately, to have a healthy immune system, and now we're kind of talking more about the second and third lines a little more specifically, we have to have cells that are constantly surveying the body, so surveillance. We have literally billions and billions and billions of cells that do nothing but sit around in all areas of the body just waiting for something to come across their path. And that's the idea of surveillance. Then, if a pathogen or a threat does come across that line, they have to have some way of recognizing it. They don't have a brain, they have little cell receptors. So you can kind of see that here in this part of the figure. These little receptors, like I drew, can lock on to different parts of a pathogen. And it's basically another example of a lock and key kind of interaction. Not quite the same, but similar. Once that interaction occurs, that cell will then be triggered to actually swallow the microbe, bring it inside of its cell, and then digest it and kill it. And if that by itself eliminates the threat, then the process is over. If, however, that's not sufficient, then the third line will be activated when the threat is more significant. So surveillance, constantly around, recognition, interaction with the receptor, and then destruction. Here you see how the microbe can be dealt with in at least one example by swallowing it and killing it. So while there's many other details, that kind of gives you a general idea of what the body is doing at kind of a basic level. Now, surveillance is, in a little more detail here, this idea that the immune cells are present and many of them are kind of moving throughout the body. And we refer to these collectively as white blood cells. And there are many different kinds, but collectively they're referred to as white blood cells. These are especially prevalent around the portals of entry. So the lungs, the gastrointestinal tract, in places below the skin where microbes can breach through. You tend to see lots of cells in those areas in particular. Recognition tends to be described like a lock and key method. It's a little broader in the specificity, but same general idea. You have a receptor, it has to bind to a specific little structure, which is part of a microbe, and you have different receptors that all have the ability to lock on to different parts of a microbe. So some parts of a microbe are lipid, you'll have a receptor to bind to that. Some microbes have flagella, you have a receptor that binds to the flagella. Some microbes most bacteria have cell wall. Peptidoglycan has the cell wall component. It has a receptor to bind to the cell wall. So the truth is, you don't just have one kind of receptor, you have actually hundreds, and these are just a few examples. And those receptors can all bind to different parts of a microbe. So it's a lock and key with many different locks and many different keys. So the receptors act as the lock, and this also applies to antibodies. And that'll come up in the next part next week when we do chapter 16. But same general idea. And the antigen, sometimes called an immunogen, acts like the key, like the peptidoglycan that fits into that little receptor with a certain degree of specificity. Once these receptors are activated, that then sends a signal to the DNA. The genetic code is then changed and the expression of DNA changes. And as a result, it activates a response mechanism or a destruction pathway that then causes the white blood cell to respond by bringing the microbe or parts of the microbe in and then digesting it or eliminating it. And if that doesn't work, then they go and activate the B and T cells for further help. Now, one of the key issues with responding to a foreign pathogen is, as I mentioned, these white blood cells, they are not cells with a brain. So they are cells that kind of feel their way through the world. And the receptor doesn't have a problem-solving brain associated with it. So if it binds to something, it reacts to it. And that's a key distinction, because what that means is that we could theoretically respond to ourself. Our own body cells contain lots of little particles that these cells could react with. And we have to then be able to differentiate between what we consider the self, our own body, and the non-self, generally speaking, things that are foreign to us. And so this is a key thing that has to take place within the body. So if we start reacting to our own cells, we will end up getting what's called an autoimmune condition. And you may be familiar with that. Autoimmunity basically means a reaction to the self. And that does happen. unfortunately. So it's not a perfect system, but generally we don't do that. And when we do, it's some kind of breakdown in the process. So any of the molecules in our body or molecules at large in the world could be reacted to. And so ultimately, there has to be a more specific way of differentiating what the microbe, what the white blood cell is responding to, so that we don't respond to the wrong kinds of cells. So how do we distinguish? Part of the concept of innate immunity is that you're born with the ability to distinguish what is foreign from what is self. And the way that you do that is that these cells contain a specific kind of receptor called a pattern recognition receptor. And these are abbreviated PRR. And a PRR is a whole series of different pattern recognition receptors that respond to an entire series of what are called PAMPs, pathogen-associated molecular patterns, kind of a big acronym. But the idea is that microbes that are foreign to us have distinct characteristics, like flagella, like peptidoglycan. And those are distinctly different from our own body characteristics. So these pattern recognition receptors have specificity to structures that are uniquely foreign, and they should not ever be found as a part of the normal body construct. So by having this specific pattern recognition series for specific molecular patterns, that kind of takes care of a certain number of potential foreign interactions. Theoretically, only a foreign part should bind to that pattern and not something from the self. So these pattern recognition receptors are genetically determined. You're born with the ability to create them, and the ability then for them to differentiate is innate. And that differentiation is based on the fact that the kinds of patterns that they recognize are those that are completely distinct to microbial cells. So for example, peptidoglycan I mentioned on the previous slide there, that is only found in bacteria. It is not a pattern that is found in the normal human body. So if you find peptidoglycan, you know, at least from an innate standpoint, that that is something that has to be foreign. And therefore the body can respond to it with a pretty high degree of confidence that that's not something that would normally be there. And so that's kind of the idea of a pathogen-associated molecular pattern. They're distinctly foreign, and there's no real overlap between the microbial world and the human world. Now, if your body responded to something like a DNA molecule, that would be a problem, because DNA is found in all life, including our own body cells. And so that would be an issue, because if you recognize just DNA as a general construct, you would theoretically start reacting to your body's own DNA and not just something foreign. There's no real difference in a general sense between the microbial DNA and our own human DNA. So these pattern recognition receptors have to pick up on things that are distinctly foreign and don't have any kind of significant degree of overlap between our own body and their microbial patterns. Now, there is some middle ground, and I'll only mention this briefly because this could be a whole long discussion. But part of what we have to distinguish here are not only just completely foreign and self, but sort of middle ground with microbes that are friendly to our body. Okay, so I mentioned this in the last week's lecture on how the body is trained in the early months of our life. Months and years, in the first couple of years after we're born. And this is a new area of immunology and microbiology, but what it looks like right now is that the microbes that are present when you're first born, quote unquote, train the immune system. And this is a layer of complexity that we won't fully get into, but the immune cells have different kinds of reaction patterns early in your life than they do when you're an adult later on. And so early on, these receptor patterns are kind of being trained, quote unquote, to make a differentiation between microbes that are there early versus microbes that might come along later as we develop into our adult years. So the bacteria in our gut, for example, appear to be tolerated if they're there from an early time period. And this could also relate to certain foods, because food could also be theoretically a foreign pathogen if your body recognized it. So things like food, things like friendly bacteria, our immune system is seemingly trained to kind of avoid those at an early age. And this can relate to how introduction of foods early could shape the immune system. For a certain period of time, it was warned to parents not to give your kids peanuts when they're young. And the reason was because some kids have peanut allergies, and it can kill them. So there's a good reason, they thought, to say, hey, do not give your kids peanuts until they're a couple years old, and then kind of give them just a little bit to see if they're going to have a reaction, and then if they don't, then you can go ahead and give them peanuts. And now that advice has kind of changed, and it's sort of nuanced because some kids can have a reaction. But what they have found is that if you give a kid peanuts early, they tend not to have a peanut allergy later. And if you wait and you give them peanuts only after a couple years, then they're actually more likely to have a peanut allergy, right? And it's tricky because every kid is different, but that's generally what they're finding, is that if you're exposed to those early, you're actually more likely to tolerate them and less likely to have a reaction. So that kind of contradicts the previous advice, keep them away from your children, because the thought was they might have a reaction and we want to avoid that. So these kind of things sort of highlight this idea that there's different periods of development in the immune system. And the idea that something is just distinctly good or distinctly bad, it's never quite that simple. Okay? And I won't go into any more detail on that here. But that's just a little kind of note about the middle ground, as I say here, where generally things are either foreign or not foreign, but that middle ground is where it gets tricky, and that's just an example to kind of give you an idea of how that might work. Okay, next we're going to talk about how the body works in terms of compartments or different areas of the body that make up the immune system. So the body consists of many different parts, and the different parts kind of make up this quote-unquote environment. So you have kind of the broad environment and then specific environments within different parts of the body. So one area would be extracellular fluid, ECF. And this is found in the spaces in between cells. So you have the internal environment of a cell, and then you have kind of an external environment around the cells. And that's one area of an interacting space, environment, extracellular fluid. Another would be the bloodstream. Generally speaking, the circulatory system. That's another area. Another important one is the lymphatic system. And although the lymphatic system does a couple key things, one of the things that it really specializes in is in transporting white blood cells, and not only transporting but housing and storing white blood cells. In fact, the lymph node that I drew in that little illustration is part of the lymphatic system. So the lymph nodes are connected to little vessels that transport lymph fluid, for example, and those kind of make up parts of the lymphatic system collectively. And these then can lead to other vessels that connect back to the bloodstream or into the circulatory system. So no environment is fully isolated. They overlap at some point. Another one that's a little more technical is called the mononuclear phagocyte system. And this is really kind of a series of connective tissues that tend to be in the area of the extracellular fluid, although in certain areas more so than others. And it actually acts like a little transport network for phagocytes. And so phagocytes, as we'll see here in the next lecture or two, are kind of the cells that swallow and digest microbes. So it kind of gives them a space to move about. It's almost like a little network of roads for the phagocytes. So they can kind of travel within that space in between one body compartment and the next. And along these little fibers, they can actually crawl and move around, traveling from one part of the body to another. So those are some of the big kind of parts or organs and cells participating in the immune system. Now here you can see in this figure the lymphatic system. So all these green little vessels represent kind of all the different areas where the lymphatic system goes. Here you can see these little circles represent lymph nodes. Over here you can see the circulatory system kind of is based around the lymphatic system, but is distinctly different. And what you see here is that circulation kind of follows both the circulatory system and the lymphatic system ultimately kind of drains back into that. So there's ways in which these overlap and interact in different parts there. Here we see a lymph node. So this would be where the lymph fluid drains into a lymph node, kind of a larger little cavity. That's what I was drawing there earlier. So they little passageways kind of move in. Cells can accumulate and sort of stay in these lymph nodes, and then if they're activated, they can travel out some of these little vessels. Actually, this is where they come in, and then they travel out to different areas of the body to kind of move about from one part to the next. So it's kind of a housing center for certain types of lymphocytes, white blood cells, and then it also gives them passageway in and out. So that's an illustration, and this is a real cross-section microscope image of what that looks like. And then here we see an illustration of the mononuclear phagocyte system. And then the pink areas here, it's shaded for where these are most dense. And what you see here are cells, the purple representing the nucleus, this being the boundary. And these little fibers here, those are the reticular fibers, of which the macrophages, things like neutrophils, macrophages, and dendritic cells, all different types of phagocytes, can actually crawl and move along the outside of the cells from one space to the next. It's almost like a little highway that these cells travel around in. And so that's kind of where the extracellular fluid and the mononuclear phagocyte system overlap. So this could be thought of as an extra outside cellular environment, and then these particular areas have these fibers for the macrophages and phagocytes to crawl upon. This is a real picture here of a certain kind of macrophage. These are the bacteria in purple or pink that are being phagocytized. So that's actually a real image, interestingly. Kind of hard to tell exactly what's going on there, but this whole thing here is the macrophage, and this right here is the bacteria being swallowed. And then this is just another figure kind of illustrating different views of how those cells look. This kind of is more like what I was showing earlier. This is the outer layer of skin, and these are the cells kind of below the layer of the skin there. So that kind of gives you an idea of the layout and some of the cells and how they move about and start to interact. The lymphatic system is a key part of the immune system, so we're going to go through that here next. The lymphatic system is a system of vessels and organs that serve as sites for the development of immune cells and then immune reactions. So that is one of the key aspects of the lymphatic system. Some of the major functions include draining excess fluid from the body and returning it to the bloodstream. This occurs every day under normal wear and tear, but is also really important when you get swelling due to inflammation. As the body gets inflamed, it tends to swell. That's actually the result of excess fluid, and that swelling eventually goes back down as the fluid drains back into the lymphatic system. It also acts like a filter, especially those lymph nodes. You can almost think of those lymph nodes like a little microbial filter. And as the body is exposed to various microbes like bacteria and toxins that come along not only with microbes but for other reasons, the lymph nodes and the lymphatic system in general kind of filter those out. It's also a place where a lot of white blood cells are produced or housed and circulate back and forth through different parts of the lymphatic system. So you kind of have multiple different things happening just within the lymphatic system by itself. The fluid that moves through the lymphatic system is called lymph. And this is similar to blood, not exactly, but it contains similar components like the plasma. If you're familiar with blood, you may be familiar with blood plasma. It's basically everything in the blood except for the cells. Basically, if you take all the cells out of the blood, you're largely left with what's called plasma. So it's kind of like that. And these lymph nodes, lymph vessels, all kind of move that plasma around and kind of squeeze it through little tiny areas called capillaries, which then drain into the lymph nodes. So within that lymph fluid, we have things like white blood cells. We have, from a nutritional standpoint, this is where fats are often filtered through the body for digestion. Any kind of cellular debris, that could be our own cells that kind of die and get worn out, or could be microbial cells that are being destroyed. And infectious agents like bacteria and viruses and fungi kind of move through this filtered lymphatic region and get kind of screened out and eliminated within that area. And so you can see here how this connects to the circulatory system. The red shows where your arteries are moving, starting from the heart. The heart pumps blood through the arteries. That travels throughout the body, and then it starts to merge with capillaries. Capillaries are little tiny vessels that form at the micro level of the tissue. The arterial capillaries migrate and sort of transition into the venous capillaries, shown here in blue. This is also where oxygen exchange takes place. So the oxygen in your blood diffuses out, is picked up by your cells. It loses oxygen. It now becomes deoxygenated, which is represented by the blue. And that low oxygen blood goes back to the heart, picks up oxygen from the lungs, and migrates it through other areas of the body. And then the lymph vessels are sort of integrated within this area, and the excess fluid that comes out here can be picked up by the lymph vessels, transported back through various filters all throughout the body, and eventually dumped back into the venous cavity near the heart to keep that fluid in circulation. And so here you see kind of what that looks like at the micro level. You have kind of the oxygenated blood coming in, and then you have some of that moving into the tissue. The fluid gets picked up here by the lymph capillaries and then migrates sort of back towards the heart over time. So really interesting but complex system that all kind of works together, not only just for microbial interactions, but also for regular circulation as well. So lymphatic vessels are a key part of the lymphatic system. These start out as those little tiny capillaries at the micro level in the tissue, and then transition into larger vessels that move back towards the heart. And so since these actually begin at the smallest level, they tend to be in high concentrations, very often near the periphery of our body. Not always, but like for example, you tend to have the highest concentration near your hands and your feet. And then another common place is near the areola of the breast. And these are not only just areas that kind of are at the periphery of the body, but they're also areas where you tend to have a lot of microbial invasion. It's more likely that you'll prick your hand or your feet to get infected, and breast can also be an area of infection, especially if you're breastfeeding. So those areas are not only kind of at the periphery to bring the fluid back to the center, but also represent areas where you have a higher chance of a microbial invasion. So unlike the circulatory system, lymph only moves in one direction. So it starts at the tissue level and then drains towards the venous system, which then migrates back into the veins and then gets dumped back into the heart. So any lost fluid, any excess fluid that diffuses out of the capillaries can be picked up and dumped back into the circulation. So it kind of allows almost like a spillover. Any lost fluid can be regained back into circulation through this mechanism here. So it tends to travel from the extremities towards the heart. lost fluid is pumped back into the bloodstream, and ultimately it not only acts as a good way to kind of filter microbes from the periphery, but to also return lost fluid back into circulation. Now, an interesting thing that's good to know about this is that the movement of lymph fluid throughout the body is dependent upon skeletal muscle, meaning just body movement in general. So if you're sitting all day long or lying in a bed for too long, what will happen is the amount of lymph flow starts to decrease. And what that means is that the excess fluid that's draining out here stays in that area. And that's when you start to get swelling. And you see this most commonly with people who have sedentary lifestyles, or if someone's in a hospital bed and they can't move, for whatever reason, they're stuck in a hospital bed, and you'll start to get things like bed sores and swelling and edema. And the reason is you need to actually move your body to get that fluid pumping. It's dependent upon muscle movement, skeletal muscle contraction. So that's one of the things that happens when you sit for too long. It's also one of the reasons why exercise is good for you. It just kind of naturally keeps this flow going in the way that it's intended to. This is also one reason why compression sleeves can be useful. So if someone can't move for whatever reason, you can compress the skin or around the body, and that kind of artificially pumps some of that fluid back in for you. It doesn't work as well as body movement does, but it does make some difference there. So that's the lymphatic vessels and capillaries. Now, in addition, these capillaries and vessels ultimately kind of migrate in and out of lymphatic organs. So you have sort of two general categories here, primary and secondary. So primary lymphatic organs are those where your immune cells develop and mature over time. So these include the bone marrow and a specific organ called the thymus. And it's easy to mix that one up with the thyroid because they actually sound similar and they're both located almost right on top of each other. So it's easy to mix those two up. But the thymus and the thyroid are two different organs. The thymus is a part of the lymphatic system. Secondary organs have more to do with the housing and the kind of long-term storage of the white blood cells. So a big one here is the spleen. You don't often think about your spleen, but it's a very important organ for holding on to white blood cells once they've been produced. The spleen is also good for filtering the blood as well. Lymph nodes, these are located all throughout the body. I mentioned those a few times here. That's a key type of lymphatic organ. There are many, many, many lymph nodes, but collectively they can be thought of as a secondary lymphoid organ. And another one is much more nuanced. Collectively, these are referred to as associated lymphoid tissues. And then you get into specific subtypes like SALT, for example. SALT stands for skin-associated lymphoid tissue. And so each of these have kind of their own little place in the body, but they kind of have a specific location that houses and reacts to microbes, houses white blood cells and reacts to various types of foreign threats. So skin, mucus, gastrointestinal, tonsils are also part of the secondary lymphoid system. That's one of the reasons why today you don't tend to take those out. Back in the day, they would take your tonsils out pretty quickly. Today, they tend to leave those in unless they get really serious. They're a key part of the immune system. Peyer's patches are found in the gut. Those are another more specific example. But those are all nuanced. We won't get into too much detail on those. But those are also important in their own little nuanced area. So in general, the kind of key difference between primary and secondary is primary organs kind of do more of development and maturation of immune cells, while the secondary organs are more for the housing and the areas where the response against pathogens tends to occur. So kind of develop and maturate them, send them out into the body, and wait for an immune reaction to occur in the secondary organs. And that's why you tend to see so many specific associated lymphoid tissues that each kind of have their own place in the body. Lymph nodes are also all throughout the body, because in any area of the body a microbe could come in. So you have all these different areas where they're kind of sitting and waiting for that reaction to occur. Now, next we're going to talk about some of the blood cells, and then we'll wrap this up here at the end with just a quick note on kind of how these cells can communicate. So there are a number of different blood cells here. So this is going to end up being a certain amount of memorization, and this can be tricky. But we'll start here with a broader term called hematopoiesis. Hematopoiesis is essentially the process by which blood cells are produced. These begin as what's called a stem cell, or more specifically a pluripotent stem cell. It's kind of a broad form of a stem cell. Stem cell is almost like a blank piece of paper, but you haven't written anything on it yet. So it kind of holds the ability to be filled in with what you need. And so you start off with these kind of blank pieces of paper, and then the body develops those over time into specific cells by kind of filling in the details. So some of these become leukocytes, still a fairly broad category. Here you can see a stem cell. Here you can see a leukocyte, broad term. And then as these develop more specifically, they develop into any of these different subtypes here, depending on exactly the way the cell is developed. So leukocytes, white blood cells. Non-leukocytes would be things like red blood cells and platelets primarily. So those are formed from hematopoiesis but end up becoming a different kind of cell, not necessarily involved in the immune system quite as much. Scientists, when they first put these terms together, this is well over 100 years ago, they didn't have all the advanced technology that we have today, but they did have microscopes. And when they're looking at the cells, they could sort of differentiate them in two broad ways, and it was based upon the grainy appearance that the cells had under the microscope. So if they had a grainy kind of appearance, they call these granulocytes, and that made sense at the time. And so you see that in this figure over here, and you can kind of see, based on the illustration, there's little grainy characteristics in each one. So they call those granulocytes. And really, it's just a way to kind of differentiate them with the microscope. And then agranulocytes are those that didn't have the grainy appearance, and that's where they got their name. So the naming is a little arbitrary, but that made sense based upon the way they saw them under the microscope. So today, you know, those terms still kind of hold relevance in terms of how they're named, and they does a decent job of grouping them together, because the grainy appearance does indicate what they do to a certain extent, but that's really where the name comes from. So when you look at the granulocytes, one of the most common cells of the immune system is called a neutrophil, and this is an example of a granulocyte. Neutrophils are one of the most important cells as a first responder to infection. So these are a key cell in the innate immune response. So they respond kind of generically to foreign pathogens, and they tend to have a really high ability to recognize foreign pathogens and then to phagocytize them, meaning to swallow them and kill them. And these are especially important against things like bacteria. If you have a bacterial infection, one of the things you'll notice if you do a blood test is you'll see an elevated amount of neutrophils. Basophils and then eosinophils are a little more nuanced, but these cells tend to be involved in more non-infectious kind of reactions, especially those going outside of bacteria and viruses. So basophils, for example, tend to release things like histamine, and they play a role in driving inflammation. And one of the things that happens if you have an allergy, for example, is you have a large basophil response. And so basophils release histamine, which is what drives, one of the things that drives inflammation. And that's one of the reasons why taking an antihistamine like Benadryl, diphenhydramine, that helps alleviate the symptoms by suppressing the effect of histamine. Eosinophils are even more nuanced. They also play a role in releasing inflammatory chemicals, but these cells are important if you have a parasitic infection, meaning something like a worm infection, which is not common, especially in America today. But if you were to have some kind of tapeworm or roundworm infection, you would actually see a significant increase in eosinophil response. Those would proliferate and migrate to the response to kill the worm. And we don't see worm infections, at least not in humans, as much, but those cells are there and they play other roles in allergies and things like that as well. Mast cells are actually very similar to basophils. One of the big differences is that mast cells tend to stay put and basophils tend to move around. At one point, they didn't even realize there was a difference between the two. They actually they seem very similar, but the large difference is that they tend to stick in one area, whereas basophils tend to move around the body. Non-leukocytes, kind of lumping these in here with the granulocytes, these would be your red blood cells and your platelets. Now, platelets generally are recognized to play a role in blood clotting, but they can overlap in the immune system where they do things like binding to bacteria or facilitating inflammation. So for the most part, we think of platelets as blood clotting, which is for good reason. That's their primary role, but they do actually have secondary roles in things like bacterial infection as well. Agranulocytes, these include things like monocytes and macrophages. These are actually almost like the same cell at different points of development. You can almost think of a monocyte something like a toddler or a teenager, and a macrophage more like an adult or a geriatric. Sort of the same thing at different points of development. So monocytes are cells, these cells start out as monocytes, and they start out by circulating in the blood. And under certain conditions, they will then develop into macrophages and sometimes dendritic cells, and then they will migrate into the tissue outside of the circulation. Once they get into the tissue outside the bloodstream, they develop into more specific characteristics, and at that point you call it a macrophage. Both these cells, kind of like a neutrophil, they specialize in phagocytosis. So generally when you're talking about phagocytes, which we'll talk about next time, you're kind of talking about things like neutrophils, monocytes, and macrophages. Even though they're different cells, they all kind of specialize in phagocytosis. Dendritic cells, another variation of these, are more specialized in what's called antigen presenting. So when I mentioned earlier that cells migrate to the lymph node, dendritic cells specialize in this. So they do what's called antigen presentation or antigen presenting. And I won't go into that detail here because that will come up later in a different lecture. Lymphocytes, these are the cells that make up the third line of the immune system, your acquired or adaptive. These include T cells and B cells, largely. There is another category here that are referred to as natural killer cells. And those are really interesting, but with the amount of time that we already spend on this, I don't feel like I have enough time to go into the kind of in-depth that those need to be fully explained. So I'll kind of mention them and not really go into the detail here. But if you're interested, these are actually really fascinating little cells. These cells do a whole variety of things that are kind of hard to summarize. But one of the things that they do is they actually remove tumor cells, cancer, actually. So your body will naturally develop cancer. And these cells have the ability to remove cancer before it ever develops into a serious form. So all these constitute what are called lymphocytes. So these will be brought up in more detail when we get into chapter 16 next week on the acquired or adaptive immune system. Last thing here, and we'll wrap this up. All these cells have to have the ability to communicate with each other. And really all cells of all kinds do some form of communication. So this is kind of like a universal concept when it comes to cells in general. But when it comes to immune cells, they use special kind of chemical messengers called cytokines. So cytokines are a specific cellular messenger that relates to how the immune system communicates. So these get real complicated, but ultimately they do a handful of key things. They direct the development of cells. So when cells need to grow, and so for example, when they're turning from a stem cell into a specific kind of granulocyte or agranulocyte or lymphocyte, the reason why they turn from one to another is based upon the specific kinds of cytokines that they get introduced to. So that kind of gives a developmental program to tell the cell what kind of cell it needs to come into being. Another way that these are used is with inflammation. So when you start having inflammation, what's happening at the micro level is cytokines are driving excess fluid accumulation in the body. And in a broad sense, they play a role in shaping the immune system. So when the body needs to attack, they tell the body when to attack. When the body needs to withdraw, they tell the body when to withdraw. So there's sort of a attack mode and a withdrawal mode, broadly speaking, that can be dictated by these cytokines. So, for example, some of these are pro-inflammatory. Cytokines that cause an engagement are pro-inflammatory. Some of these are anti-inflammatory. They sort of cause a withdrawal from the engagement. Some of these affect blood cell permeability, causing, for example, constriction. Blood cells can be induced to contract and get more narrow. They can also be induced to dilate and get more wide. And then another one is that they can cause the stimulation of growth factor cytokines. This is kind of where the cell development kind of comes in. So they can stimulate, especially during the hematopoiesis, which cell you're supposed to turn into. Okay? And this table here is from the book, and this kind of gives you some examples. I don't test you over memorizing all of these. There's already enough to keep track of. But these give you some examples of what kind of specific cytokines are involved, where they come from, and what they do. So if you want to kind of know more detail, you can take a look at this chart, and there's many other charts online that kind of give you some examples of what those do. All right, so we'll wrap it up there, and next time we'll take a look at the innate immune system in a little bit more detail, kind of honing in on the first and second lines of defense.